Hello students, welcome to Turito live classes. Myself A. Swagat. Today in this particular session, we are going to understand about a new topic, that is urine formation. So, in this particular session, we are going to look into the actual functioning of the nephron, how does it function to form the urine. So, that is what we will begin with. So, before we do that, we will be looking into the topics what we have already completed in the previous session. So, in the previous session, we recalled the excretory system in human beings. We tried to look at the kidneys and its internal structures and how the nephrons are arranged within the kidneys, and finally, that is how we came to the arrival of anatomy of human kidneys. So, that is how we ended that particular topic, following which we looked at the nephron, that is the structural and functional unit of kidneys, followed by we had a detailed analysis of the nephrons, that is the structural analysis. We try to understand each and every part of the nephron, how it is formed and what does it have, its functioning. So, that is what we did, following which we also understood the classification of the nephrons, that is cortical nephrons and juxtamedullary nephrons. There were two nephrons, we looked at it, and there is a lot of difference between them. It is all about the size of the Henle's loop. So, today in this particular session, we are going to have the understanding about how urine is formed in the body. So, about which we are going to look at the objectives starting with this. So, we will be listing out various steps involved in the urine formation process. After that, we will look into the functioning of the glomerular filtrate. So, in the nephron, we have seen the Malpighian corpuscles or renal corpuscle. So, that is a structure which is, we know it as Bowman's capsule holding the network of arteries. Do you remember that? So, our story for today's session would start from that Bowman's capsule and the network of the arteries. So, that is where we will see it forms the glomerular filtrate, and we will also have a detailed view of the renal corpuscle, following which we will look at the functioning of the PCT and the descending loop of Henle. So, children, can anyone tell me what do you mean by PCT? Expand PCT. Correct. So, it is quite simple, it is proximal convoluted tubule, that is what is PCT, and then we will be looking at the substances that are filtered. Okay. Which of the substances are filtered? So, we have a specific name for that known as threshold substances, understood. So, we will be looking into them, and following which we will understand the formation of the hypertonic urine. Hypertonic urine means it is the concentrated urine. So, that is how it is forming. So, how it is forming? So, we will look into the functioning of each and every part of the nephron, and that will give us an understanding about it. And when we say hypertonic urine, it is the urine where you will find that the solute concentration is more as compared to the solvent concentration, thereby making the urine hypertonic. After that, we will also look into a process called Micturition, which is actually the process how manually urine is released out of the body. So, that is what we have planned for this particular session. These are the following objectives for the session. So, let us move forward and let us understand it. Let us see now. Okay, here. So, human excretory system. So, when we think of human excretory system, we will understand that. So, in this we have 4 steps. So, we will be understanding the same thing with the 4 steps here. So, this is the first step, okay, glomerular filtration. Second step is selective reabsorption, selectively how reabsorption occurs. Third is tubular secretions, that means something is given out of the tubes of the nephron. final step is formation of the hypertonic urine. So, you see glomerular filtrate, what can you make out from this? So, glomerular filtration. So, okay. So, it is quite simple. I see some of the children dropping in the response based on your understanding, and you read here it is mentioned as glomerular filtration. So, we have the glomerulus which is inside the Bowman's capsule, that is where exactly the first step of urine formation begins. Okay. That is what we will be looking into. Okay. Now, here what happens in the glomerular filtration. Okay. So, we have seen these are the 4 steps. I will just give you a brief insight into the steps. Okay. After that, we will go into the details of each step separately. Okay. So, that is how we have planned for it. Okay. So, let us begin. So, glomerular filtration, if you see here, in the glomerulus, which is present inside the Bowman's capsule, okay, that is where you will see the ultrafiltration of blood happens. So, ultrafiltration of the blood happens. Okay, that is what happens. Now, what you are supposed to understand here is, when you look here about the ultrafiltration is happening, that is where every ion, every atom, or every molecule which is going through that filter would be filtered, and everything will be filtered, right? Good or bad things, everything is filtered. So, there is a reason. You might ask why there is a need to filter everything. Now, that is where the trick lies. It is in the glomerular state where it is completely filtered, okay, following which you will see second step, selective reabsorption. Here everything is filtered out. Okay. Second step, you see it is mentioned as selective reabsorption. So, when the substances are passing to the other tube of the nephron, those tubes, the other tubes other than the Bowman's capsule, okay, I am speaking about PCT. So, you see there happens the process which is beginning, that is selective reabsorption, because children, when you think of the nephron, it is only the Bowman's capsule and the glomerular which is present inside the Bowman's capsule is designed to filter, and that is the only system in that nephron which has the ability to filter very well. So, that is why you see ultrafiltration occurs there. So, it filters everything, okay, good or bad things which are required by the body, which are not required by the body, everything is filtered there. Now, when everything is filtered there comes a second step, because we have filtered those which are needed by our body also, right. That is why we have the second step for selective reabsorption. So, selectively it will reabsorb the filtrate what is already given out. So, in that selective reabsorption, those substances which are required by the body would be taken back into the system. Okay. After which you will see the third step, that is tubular secretion. So, you have that Henle's loop, okay, and we have what you call there is one more tube called DCT, distal convoluted tubule. So, they all are functioning in a way. Now, here what happens, this tubular secretion means few substances would be given into the filtrate, that means they are secreted by the tubes, most probably they are potassium ions, sodium ions, okay, they all are secreted there. Why it is secreted there? Because the moment when you remove something from the system, it definitely alters the nature of the fluid, right. I mean the acidity, basicity, that will alter the system, is not it. So, in order to avoid that alteration and to make the fluid, it should be having a specific pH that has to be maintained. In order to maintain that pH, tubular secretions happen, that means inside the tube few substances are secreted by the tube, so that that balance, that osmotic balance would be maintained. So, which also involves the maintenance of the concentration of the substances in the fluid, which will also account for the maintenance of the pH of the fluid. After that, you will end up in the last process of formation of hypertonic urine. So, you will end up having the process of hypertonic urine. So, that is more important for us. Okay. Now, this is how happens. This is what the brief understanding of the urine formation process. Now, what we will do is we will move forward and we will look at it how different steps are, we will analyze each and every step carefully. Okay. Let us begin here. So, first thing is glomerular filtration. So, let us see here what we have. So, filtration of the blood, you see here. So, like I mentioned, filtration of the blood happens, okay, and following which you will see we will have the filtrate contains almost all the constituents of the plasma except the proteins. Like I mentioned, everything is filtered, but we should not filter protein. So, that is not filtered there, that is how it is designed. So, it is done by glomerular filtrate. Okay. So, glomerular filtration. So, whatever is filtered is what is filtrate, and that filtrate contains all the constituents of the plasma. Whatever is present inside our plasma, we get to see that in that particular filtrate. But when it comes to proteins, you will not find any proteins in that system. Okay. Fine. Next comes up, we have the filtrate thus formed is called ultrafiltrate. That means, leaving proteins, whatever is present inside that is known as ultrafiltrate. Okay. So, which is also known as glomerular filtrate or primary urine. So, the substances which are filtered and you get to see them in that filtrate, that is whatever is filtered, that will be left out, right. So, inside the filter, that is what it is thinking of. Okay. Now, when you see here, we call that as ultrafiltrate, or it has another name, that is glomerular filtrate, which is also known as primary urine. Okay. So, we do not release this primary urine. Why do you think we do not release this primary urine? Correct. So, some of you are putting the correct answers here. We do not release the primary urine because it contains almost everything which is present in the plasma. Certainly, we cannot, our body cannot afford to lose all of them. Certainly, we do not use that. So, this primary urine has to undergo several processes and it has to be treated properly inside the system. That is when you see that it will be released outside. Now, you see this primary urine is a hypotonic to the cortical fluid. Hypotonic means it is having lot of fluids, less of solute, right. So, that hypotonicity is with respect to the fluid present in the cortex region of the kidney. Got it? So, that is why it is mentioned here as cortical fluid. So, this hypotonicity of the primary urine is with respect to the fluid present in the cortex region of the kidney. So, this is what is mentioned here as cortex. Okay. So, it passes into the next part of the renal tubule. So, that is what we are here to look at. Now, when you think of the renal tubule, you will see that it will be having the amount of filtrate found by the kidneys per minute is called glomerular filtration rate. So, what you see here is the amount of fluid passing through each and every every time, whenever the fluid passes through the glomerular filtrate, it has to filter this much amount of fluid per minute. that rate is mentioned here as glomerular filtration rate, which is also known as GFR. So, that is mentioned here in short, that is GFR. Okay. So, hope you got it. Now, so GFR in a healthy individual is approximately 125 ml per minute and accounts for 180 liters per day. I mean, this is the volume of fluid that is getting filtered in our body per day. So, that is for one day. So, I am sure many of you might be confused looking here, 180 liters of substance is there in our body or what? Certainly we do not have that, children. We certainly do not have 180 liters of fluids in our body. So, you might be thinking then why it is mentioned 125 ml per minute and that accounting for 180 liters per day, that might be your confusion. Well, I will tell you, it is said that 125 ml of the fluid is filtered per minute. Okay. So, let us think this way. For example, okay, we have, okay, to use some other color for you. Okay, for example, we have 100 units of fluid, let us say. Okay. For example, we have 100 units of fluid in our body. Okay. Certainly, that is less than the 180 ml what is shown here. Okay. Now, what is done here is, it says that 125 ml out of that 100 units of fluid passes through the glomerular, right, that too per minute. That means, first set of 125 ml pass through that filtration system and it passes, then the second set comes, then the third set, it is a continuous process. Now, the first set which already filtered earlier will again would be visiting the filtration system in the Bowman's capsule and that will be getting filtered again. So, hope you understood that. So, likewise, that first set what I said is 125 ml, second 125 ml for second minute, okay, then third 125 ml for the third minute. So, that happens continuously. Okay. So, ml for third minute, ml, milliliters. So, first 125 ml of fluid which has passed through the glomerular filtration system, it will again pass on through it once it is going into the body fluids, because formation of the urine, formation of the metabolic wastes is a continuous process, because of which our body fluids would have the wastes, that is metabolic wastes generated continuously and they are present in the fluids. So, that is why it is mentioned here as 125 ml per minute is the speed or the rate, and having 180 liters per day of that amount of fluid is passing through the glomerular filtration. Okay. The glomerulus which is present inside the Bowman's capsule, that is what it is saying. Okay. So, hope you understood that. Let us move forward. Okay. So, you see glomerular filtration system, it looks like this, it shows like this. So, what is mentioned here is very simple, children. So, this is a system which is actually involved in ultrafiltration of blood. So, this is what is glomerulus. Okay. So, this one actually involves in ultrafiltration of blood. Okay. So, this is where the ultrafiltration of blood takes place. Now, this glomerulus is actually a network of arteries. So, it is a network of arteries. Okay, we have to see that it is a network of arteries. Now, what happens here is, this network is formed of two different arteriole. One is efferent arteriole which is entering inside the Bowman's capsule. So, this one comes here, okay, it forms that network, that is only the artery, okay, and then you see it is again going outside. So, the arteriole which is going outside is actually the efferent arteriole. So, this is where the first step of urine formation happens, got it. So, this whole system is also known as Malpighian body or renal corpuscle. So, this is the system what we have. So, this is where the first step of urine formation begins. So, hope you understood that. Now, let us see. So, here we have selective reabsorption. The second step says that it is the selective reabsorption process here which occurs. So, the tubular epithelial cells. So, inside the tube, you know the epithelial cells are the upper lining of the cells. So, these epithelial cells, they are present on our body skin, they are present on the surface of the organs, they are also present on the blood vessels, they are also present inside the internal lining of the stomach. Same stands for internal lining of the arteries and veins, and the same stands for nephron. Even nephron have the similar structure available. So, that is why it says the tubular epithelial cells in different segments of a nephron. You know, nephron is having different segments, right? We did learn about it, right? So, they reabsorb certain substances of the glomerular filtrate. So, either by active or passive mechanism. So, I think by now you understood why there is a system given here as selective reabsorption. What is happening there? So, if you look here, this selective reabsorption is happening because there are different tubular epithelial cells are available, okay, different segments of the nephrons are available, they will be reabsorbing whatever is filtered, whatever is present in the glomerular filtrate. So, that reabsorption, it may happen free of cost, that means no energy is used, that is what is passive, you know, it will be absorbed passively, and sometimes same in order to absorb the glomerular filtrate, we need energy also to be used. So, that is why it is mentioned here as active and passive mechanism of the reabsorption process. So, you know how much? About 80% of the filtrate formed is reabsorbed. You know what does it mean? From the glomerular, what it was filtering, glomerulus was filtering the blood, right? So, it has given out 100 units of the filtrate, let us say. Out of that, 80% is again taken back into the body fluids. Okay? So, it happens in a constant way, and it is unregulated fashion, that is what we have to see here. So, by this is taking part in reabsorption process, and the other thing what is taking part is descending limb of loop of Henle. So, you have seen, so there was a loop like this, this whole loop is known as Henle's loop, okay. So, which loop is reabsorbing? It is this loop which is known as descending limb of loop of Henle, that is the one involved in reabsorption. Along with that, proximal convoluted tubule. Soon after the Bowman's capsule, you will see some tube would be entangled like this, isn't it? It is having curved structure. It is that tube along with this descending loop of this hairpin like structure, that is loop of Henle, involved in reabsorption. Okay. So, remember this PCT and descending limb of loop of Henle, okay, they are involved in doing that. So, hope you understood that. Now, what happened here? How much is to be absorbed? That is based on the necessity of reabsorption. You cannot expect always 80%. It may increase, it may decrease depending upon the fluid nature present in the blood. Okay. Now, you see the substances of glomerular filtrate can be categorized. So, everything is filtered there, okay, that filtrate contains everything except the proteins. So, depending on that, that has to be categorized, that means classification of that glomerular filtrate, that is what we will be doing. So, if you look into that detail, you will understand that there are high threshold substances and there are low threshold substances. So, this is the classification we have. Before we go through this classification, one must understand what is threshold substances. Let us go and understand now. Okay. So, here if you see, this is actually showing, right, I was telling about this is the glomerulus which is having the ultrafiltration process. Okay. So, it filters everything. So, in the moment when it is passing through this structure, that is proximal convoluted tubule, and it is passing through one more limb, that is this one. This is descending loop of Henle. These two tubular structures of the nephron would be involved in selective reabsorption process. So, selective reabsorption process done by these two structures. So, they are very much important. Now, inside these structures, that is what we will see the formation of the high threshold substances and low threshold substances. Now, we will look into the understanding what exactly are this high threshold and low threshold substances. So, threshold substances are the actual excretory substances. Okay. That is what for our understanding. So, these threshold substances you are looking here, they are actually excreted products that are not reabsorbed at all. One such example is creatinine, because we are discussing about the nephrons which are highly linked, that is the structural and functional unit of kidneys. Now, if anyone wish to have the health of the kidneys, anyone wish to understand the health of the kidneys, this is one particular substance. If anyone would undergo a blood test and say we need to have a creatinine test, based on the amount of the creatinine in your blood sample, it will indicate the health of the kidneys. So, normally if anyone would like to go for checking their health of the kidneys, one has to undergo a test, it is the blood test, which involves the content of the creatinine that indicates the health of the kidney. It should be within a range, a specified range. So, that depends on the types of analytical methods. Fine. So, you see threshold substances are actually the excretory products, which usually they should not be absorbed again, right. We do not want them in the body, so we should not absorb them. So, one such type is high threshold substances essentially and are efficiently reabsorbed which include glucose, amino acids, vitamins and some salts. These substances are filtered. So, our body needs them, so they are absorbed. Okay. Threshold substances are usually the excretory products, they should not be absorbed, but we need these are not excretory substances, right. So, these are certainly not excretory products, so we need them. So, that is why when it comes to low threshold substances, they are absorbed in very little amounts, and the example is urea and uric acid, right. Urea, uric acid and those substances which may contain ammonia, they will be reabsorbed, because these are the actual excretory products, they should not be in our body. So, that is what happens. Now, next you see here tubular secretions. So, selective reabsorption is done. You have seen which are the substances to be reabsorbed and which are the substances which are to be left in the fluid, which is to be discarded, that is urine. So, now you see tubular secretions. If you see here, so during the formation of urine, cells of the tubes, because you see the whole system is built of several tubes, PCT is one tube, loop of Henle has two tubes, and we have one more tube called distal convoluted tubule, okay, that is one more tube, and again we have a straight long tube, that is collecting duct, isn't it. So, the whole system of nephron itself is made of several tubes. So, in that we will find some of the tubes, they actually secrete some substances, they release few substances. So, if you see here, they release hydrogen ion, potassium ion and ammonium ions, okay. Even so potassium ion, ammonium ion, even sodium ions are also given out. Now, why it is to be done? It is done to maintain the basic nature of the body fluid, got it. That is what is mentioned here, is helps in maintenance of ionic and acid base balance of the body fluids. Interesting, right? In the first step you filter everything, whether good or bad, except the proteins, you filter everything, and in the second step you absorb what is required by the body, okay. And in the third step, by few substances giving out by the tubular cells, they maintain the nature of the body fluids, right. Now, you might ask me, sir, why there is a need to absorb everything, why there is a need to selectively reabsorb and again maintaining the body fluid nature? That is a very highly valid question, but the thing here is, if you observe the structure of the nephron, it is only the glomerulus present inside the Bowman's capsule that is designed for ultrafiltration, okay. That is actually designed for ultrafiltration. Leaving that part of the nephron, leaving that particular segment of the nephron, if you think of PCT, loop of Henle, DCT, they are not designed for filtration, understood. They are not designed for filtration, filtration never happens there. There only absorption happens or some substances are added into the fluids. Other than that, you will not find any sort of filtration or removal of the substances, you do not find much of that. That is why this process is followed in this manner. So, this is the tubular secretion. So, you see the nephron, I was just referring, this particular system is the only one designed for ultrafiltration. So, can you see anywhere else we have that kind of system to go for ultrafiltration? We do not have. You see the Henle's loop and even the proximal convoluted tubule is designed for reabsorption, and distal convoluted tubule is actually designed for secretion some substances, so that that will balance the body fluid nature, that is presence of the ions, presence of the body fluids, all accounting for pH of the body fluid. Ultimately, it is osmoregulation, okay. So, that is what. So, this collecting duct, this long tube is collecting duct, okay. So, they all are designed in a different manner, you see that. So, this if you look, there is formation of hypertonic urine, the final step. Now, what happens here? Let us have a look. So, when you look here, 75% of the water content of the nephric filtrate is reabsorbed. Means whatever is filtered, everything has to filtered with a common medium or the media, that is water. So, you cannot expect nephron should give out all water from the body, it is not possible. They need more water to function properly, right. So, once the filtrate is taken off, it is not that we have to give out the water, water is needed by the body, right. So, that will be reabsorbed, that is what is given here, right. That is reabsorbed in the region of the PCT, and 10% of the water passes out of the filtrate through osmosis in the loop of Henle. So, that is what this reabsorption process occurs by active or passive mechanism mentioned, right. One such passive mechanism is osmosis, where there is no energy is used and 10% of water is just given out. Otherwise, the little amount of water which passes out through the urine, you cannot expect the system to give out only the solutes, it is not possible. Everything is happening in a medium, and that is liquid medium, aqueous medium, right. So, the main constituent of the aqueous medium is water. So, using the 10% of water, the excretory substances are given out from the body, right. That is a simple process what you see here. Now, you see the further concentration of the urine takes place in the collecting tubes in the presence of hormone called vasopressin. So, you have seen that collecting tubes, that is a long tube, okay, which we call it as collecting ducts. That is where you will see concentration of the urine happens. Concentration in the sense addition of more and more solutes to the solvent passing through it. That solvent is 10% of that water, right. In that you will see that concentration of urine happens like that. Okay. So, the hormones is secreted only when concentrated urine needs to be passed. So, that vasopressin is a hormone that gives a signal to secrete the concentrated urine, that is urine which is consisting more of the solute content. That is when we call it as the concentrated solution, right. So, that is what happens. So, that is to be passed out. So, the hormone action maintains the osmotic concentration. So, the hormone action maintains the osmotic concentration of the body fluids. So, that is what is osmoregulation happens. So, this osmoregulation is very much important, right. So, osmoregulation, that is osmotic concentration of the body fluids, that occurs quite commonly. So, final step is micturation. So, how does this or what is this micturation? So, in the objectives, when we were looking at the objectives, I told how the urine is actually passed out of the body, that manual expulsion of urine. So, you see here the expulsion of urine that is stored in the urinary bladder. Obviously, urine is stored in the urinary bladder. Okay. That expulsion is known as micturition. Expulsion of urine which is stored in the urinary bladder is known as micturition. When we have and when we feel that the moment we get the urge to urinate, which means before we get the urge to urinate, our urinary bladder has to reach its maximum capacity. Normally, our urinary bladder can have a minimum capacity of 500 ml. Okay. So, it is a bag which stores the urine inside it, okay, and minimum capacity is 500 ml. Under extreme cases, okay, for an average adult human being, the minimum capacity of the urinary bladder is 500 ml. And under extreme cases, when the urinary bladder can stretch a little and it can hold some more urine, right, that amount comes to be 800 ml. That is 800 ml be the maximum amount of urine, 500, it is just 500 ml is the amount just fills the urinary bladder. Okay. Let us see what else we have for you. So, here when you observe, so the capacity of the urinary bladder is above 800 ml, okay, but we reach the maximum, the normal volume is 500 ml. So, after this 500 ml, the bladder wall starts getting stretched. Like I said, 500 ml is the maximum amount, in the sense just to fill it. After that, the urinary bladder starts stretching itself. So, the urine formed by the nephrons is ultimately carried by the urinary bladder. So, whatever is filtration we have understood, Bowman's capsule, again it goes to the PCT, then to the what is that Henle's loop, again it goes to the distal convoluted tubule, then to the collecting duct. It ultimately passes into the urinary bladder and in the form of urine. So, where it is stored till a voluntary signal is given. That means, we store the urine in our respective urinary bladder and we will only get the urge, only when we feel that, once the urinary bladder gets filled completely, that is when it sends a signal to the CNS, that is central nervous system. Okay. Then it has to come from the brain, the central nervous system, that will give a message again to release the urine, right. So, that is what happens here. So, when you see here, when we get the message to the brain, that is central nervous system are involved in there. So, the stretch receptors which are present in they generate a nerve impulse. Stretch receptors means those receptors, when they sense the stretching of the bladder, they receive the message carried by the sensory neuron to the brain. Okay. So, these sensory neurons are giving a message to the stretch receptors present in the urinary bladder, that means brain gives a signal and stretch receptors receive it. Okay. And then that is when you produce the sensation or you have the urge to urinate, that is when it happens, that is the process. Okay. It is only then you would see that yourself, we end up going to the process of urination, right. So, here we have the summary for us. So, let us have a look. So, in this session we listed out various steps involved in the urine formation, okay, followed by functioning of the glomerular filtrate, right. So, it was filtered by the glomerulus and how that filtrate was helping us to form the urine, we saw that. After that we had a detailed view of renal corpuscle, and then we looked at the reabsorption, selective reabsorption by PCT and descending limb of Henle's loop, okay, following which we also understood about understanding of the threshold substances which are filtrate, which are present in the filtrate, right. That is what we have seen. Once done, we looked at the hypertonic urine formation, which is expelled from the urinary bladder by a process called micturition, right. So, this is what we had for this particular session. Okay. So, you will be meeting with us in the upcoming session, we will discuss some more topics. Thank you all.